latest research, strategy, and tools to help you do your jobs. And thank you, and to protect food safety and public health out in Canada, across Canada. And now that we've actually gone virtual, might I say, even globally. So once again, thank you for attending and welcome. Today's seminar fits our mandate very well, and I am very happy to be serving as moderator. We have Richard Harris, who is going to be just attending just a moment. I do also, though, want to take a quick moment to thank the Guelph Food Safety Seminar Working Group, a merry band of food safety passionate people under the leadership of Dr. Susan Sabatini. We put a lot of effort into making these things happen. So again, I want to thank the entire group. I also want to thank our employers, our organizations, who provide us with the time and the support to make this happen as well. A couple of quick housekeeping things. We encourage questions, absolutely. Please put them in the chat, and we'll have time at the end for the questions. In the meantime, please keep your cameras off and keep your microphones muted. So at this point, I would like to introduce Dr. Richard Harris. Richard is a lab tech at Health Canada, working for the Botulism Reference Service for Canada with supervisor Dr. John Austin, who's recently retired. In the 3 years since working at Health Canada, Richard has authored or co-authored 9 peer-reviewed publications on the subject of botulism and represented Canada at 3 consecutive meetings of the Interagency Botulism Research Coordinating Committee. Richard's research interests are primarily concerned with discovering the source of C. botulism, botulinum, spores for cases of infant botulism. On a personal note, I first met Richard last year when he helped us extensively with guidance and support as we investigated a case within our health unit. It was such a pleasure to work with Richard on that, and it's an equal pleasure to introduce him here and this seminar to you at this time. Richard.
Thanks, Paul, for the introduction. And I do touch on that case at the end of the seminar, so I'll come back to it later. And good morning, everyone. As Paul mentioned, my name is Richard Harris from the Botulism Reference Service for Canada. We're a lab in the Bureau of Microbial Hazards of Health Canada, and we're located in Ottawa. I want to quickly thank Paul, Sumitra, and Susan, and other organizers of the Guelph Food Safety Seminar Series for the opportunity to introduce our lab and our work. We're always looking for ways to connect with people from other departments, agencies, and institutions to spread public awareness of this disease and opportunities to collaborate on future projects. So the Botulism Reference Service for Canada was established in 1974, and we have the following objectives. Primarily, we assist physicians and public health officials when botulism is suspected, and this happens on a case-by-case basis. Oftentimes, we get calls from primary care physicians or hospital admins discussing certain cases, and we offer guidance and recommendations. And then the other big thing we do is examine suspect foods and clinical specimens submitted for analysis. And so that's primarily my responsibility as the lab technician doing the testing for the toxin and the bacteria. We also rapidly alert responsible agencies when commercial foods are involved, such as CFIA and Public Health Agency. And we maintain reference cultures of neurotoxic Clostridia, which is growing every day. And finally, maintain liaison with centers that have similar responsibilities in Canada, such as the BC CDC Public Health Lab, that actually they do their own testing for botulism for the province of BC, and the National Microbiology Lab in Winnipeg that are viewed as a backup for our lab, and then also abroad. And then other institutions and agencies within Canada that we don't often get to talk to, like public health or academia or industry. So always happy to collaborate with folks. The leadership of the Botulism Reference Service has changed over the years. So it started with Claude Dolman, who performed investigations into botulism outbreaks in Canada from 1947 to 74 at UBC. And then Andres Hauschild took over the lead when it was moved to Health Canada in 1974 to 1990. And then it was Karen Dodds who took over the position. And in 1993, Hauschild and Dodds published what I consider to be the most comprehensive textbook available on the ecology and control of Clostridium botulinum available. And I encourage you to look it up if you're interested. I have a copy of it right here that I keep on my desk because it's such a good resource. And then in 1994, John Austin became the leader of the lab, and under his direction, we became certified with ISO 17025 standards with the Standards Council of Canada. And as Paul mentioned, John retired recently in October of last year. So now it's myself running the lab, as well as a fellow lab technician doing more of the research side, and that's Ryan Boone. So botulism is caused by the organism Clostridium botulinum. It's a rod-shaped, gram-positive, obligate anaerobic spore-forming bacterium. You can see the classic rod shape on the left, and below that, a nice scanning electron micrograph from a recent paper. This is actually Clostridium sporogenes, which is a close family member to botulinum. And you can see the rod-shaped bacteria emerging from the spore coat, which is shed like a winter jacket. And the spore coat protects the bacteria from things like oxidative stress or heat. Clostridium botulinum spores occur naturally in soil and marine sediments and are ubiquitous in nature. So foods are not normally tested for their presence. It's simply assumed that they could be present, and then efforts are made to prevent their growth. Clostridium botulinum causes disease by producing the botulinum toxin, also known as Botox. And this is the most potent toxin ever known. The species was originally designated based on the ability to produce the toxin, but nowadays it's known that there are 6 distinct genomic groups of toxin-producing Clostridia that don't technically need to be considered botulinum, but I will go into detail on that in the next slide. And Clostridium botulinum and the toxin are shed in stool, which still remains the best clinical specimen for testing. So these are the different groups of botulinum neurotoxin-producing Clostridia. Groups 1 and 2, highlighted in gray, are the most common, and they produce botulinum toxin types A, B, F, and B, E, F, respectively. Group 3 produces types C and D, which cause disease in animals, not humans, while groups 4, 5, and 6 are considered rare. So we tend to focus on groups 1 and 2, which have different physiological properties in 2 important areas that I've highlighted with red ovals. Group 1 has a minimum growth temperature of 10 degrees Celsius, while group 2 can actually grow in refrigerated conditions. However, group 1 spores are highly heat resistant. They have a D100 of 25 minutes, while group 2 spores have a D100 of less than 0.1 minutes, so they're heat sensitive. And given the heat resistance at boiling temperatures, a standard commercial canning process combines heat and pressure to deliver a botulinum cook, or a 12D process, that offers a substantial margin of safety by inactivating 12 logs of contaminating Clostridium botulinum. Group 1 spores are most commonly found in terrestrial soils, and so they're associated with outbreaks in things like fruits and vegetables, while group 2 spores are most commonly found in aquatic sediments, and so are associated with outbreaks in fish and marine mammals. The botulinum toxin mechanism of action is really interesting and worthy of a spotlight. So under normal circumstances, shown on the left panel, nerves that connect with muscle tissue are able to mediate synaptic transmission through the release of acetylcholine neurotransmitters. These bind to receptors on the postsynaptic membrane and facilitate the propagation of an action potential, leading to muscle contraction. This process is mediated by snare proteins that bind and tether presynaptic vesicles to the membrane. On the right panel, you can see that the botulinum toxin in black has a heavy chain and a light chain. The heavy chain is recognized by receptors on the presynaptic membrane, and that facilitates the invagination of a vesicle where the pH drops and promotes the release and translocation of the light chain into the cytosol. The light chain contains the catalytic domain of the protein, which cleaves the snare proteins and preventing them from fusing with the membrane and blocking synaptic transmission. The reason this is such a powerful mechanism is that these proteins persist inside the cell and continue to cleave their targets, which results in a prolonged effect and can result in extremely long recovery times for physical disabilities. The problem is that once the toxin is inside the cell, it can't be targeted by neutralizing antibodies of the antitoxin. So we always recommend that the antitoxin be administered as soon as possible based on a physician's diagnosis from symptoms and not to wait for lab results. Our lab uses the mouse bioassay to determine the presence of botulinum toxin and viable Clostridium botulinum in clinical samples, including serum, stool, and gastric contents, as well as food samples, including traditionally prepared indigenous foods, home-prepared foods, and commercial foods. The simplified method for performing investigations is quite simple. Once the samples arrive, they're put into sterile falcon tubes and centrifuged at a high speed. If the sample is to be investigated for viable Clostridium botulinum, such as the case of stools or food, then the pellet is used to inoculate enrichment cultures under selective pressure and incubated anaerobically for 5 days. The media is centrifuged again, and the supernatant is used for the detection of the toxin. The assumption being that if the toxin is found in the media supernatant, then the original sample contained viable Clostridium botulinum. Whether it's a direct extract from clinical or food samples or from enrichment cultures, the supernatant is filtered through a pore size that is permissive for the toxin. This filtered supernatant is then injected into the peritoneum of mice, and they're observed for up to 3 days. If the mice show no symptoms, then the sample is deemed negative for the toxin. However, if the mice develop characteristic symptoms of botulism, including pinched waist and labored breathing, then they're euthanized and the sample is presumed positive. The extract needs to be neutralized, though, with a serotype-specific antibody, and the injection is repeated. If the mice show no symptoms with the neutralized injection, then the sample is deemed positive for the toxin, and its serotype is specific to that of the neutralizing antibody. This entire procedure can take several days or weeks to complete, which is why, again, we advise physicians to administer the antitoxin to the patient based on a differential diagnosis and to not wait for lab results to come back. So today I'm discussing 2 main clinical presentations of botulism, the first being foodborne botulism, which is the classic way of thinking about botulism, and then the second is infant botulism, which has become more common in recent years. But I'm going to start by talking about foodborne botulism, which is a rare disease in Canada. It affects about 1 in 10 million people annually, and it tends to occur in outbreaks. But since it's a severe disease, 1 individual is still considered an outbreak. From 2006 to 2021, the average hospital stay was 48 days. 70% of patients required mechanical ventilation, and there were 7 deaths within this reporting period. The cause of foodborne botulism is growth of Clostridium botulinum in foods and the ingestion of the toxin. Symptoms include symmetrical descending facial paralysis affecting the eyes and eyelids, eventually progressing to difficulty talking, swallowing, and breathing as the toxin affects the throat and diaphragm. Treatments are limited to mechanical ventilation and the use of the heptavalent botulinum antitoxin. The data that I'm going to be presenting today was taken from our recent publication in Emerging Infectious Diseases. So if you're interested, you can look up this paper. And I updated the figures to include more recent years of 2022 and 23. This is the entire history of foodborne botulism in Canada, dating back to 1960 to the present, although there are some early reports dating back as far as 1919. As you can see, the number of cases per year can differ quite significantly because of the nature of the outbreaks. Some years have no cases reported, while 1985 was the year of the largest outbreak in our history, including 37 cases caused by garlic and oil at a Vancouver restaurant. I wanted to highlight the disproportionate impact this disease has on Indigenous communities, which are now overlaid in black. Over this reporting period, 75% of all cases came from outbreaks in indigenous foods, which are traditionally aged or fermented seal, walrus, or whale meat, as well as some salmon eggs. This disproportionate impact was especially true prior to the year 2000. A former PhD student under John's supervision named Daniel Leclerc did some source investigation in indigenous communities of northern Quebec. He worked directly with hunters to examine their practices for preparing the meat and did analysis on the prevalence of Clostridium botulinum in the soils of this region. He discovered that the main factor contributing to the growth of Clostridium botulinum in these foods was that they were being incubated in temperatures that were not low enough to prevent the growth of the bacteria. In the years since this work was completed, the rates of foodborne botulism in these communities has dropped significantly. And I'm going to focus the rest of the talk on the more recent history of the past 17 years. So zooming in, from 2006 to 2023, there have been 71 lab-confirmed cases of foodborne botulism from 59 outbreaks. There was an annual case rate of about 4 per year, and that is about half of the annual case rate dating back to 1960. And the annual incidence was calculated at 1.2 cases per 10 million people, which is again about half of what it was dating back to 1960. So we're trending in the right direction. And as I mentioned before, indigenous cases have made up less than half of all cases in this reporting period, which is a substantial reduction from 75% previously. But we do get some cases from indigenous communities in northern Canada, including one last year. When this is broken down by toxin serotype, you can see that most of the cases are caused by toxin type E in red at 51%. And this correlates with indigenous outbreaks, which are most commonly type E, which I'll get into later. Type A represented 24% of cases in blue, and B represented 15% of cases in orange. Then we have a few type F, representing 6% of cases in purple. In 2 instances, the serotype couldn't be identified because the sample was neutralized by a polyvalent antitoxin, and then we didn't have enough sample left over to determine the serotype. So those are considered unknown in light blue. And then interestingly, we had one case in 2021 caused by a Clostridium botulinum that produced both type A and B toxins. And that's in green. This is the geographic distribution of foodborne botulism cases in Canada from 2006 to the present. Quebec had the highest number of foodborne cases at 31, and I separated Quebec into 2 to show the proportion of cases coming from the northern peninsula of Nunavut, where there are indigenous communities that frequently have type E outbreaks, and then the rest of Quebec that show a more diverse proportion of toxin serotypes. Ontario had the next highest number of cases with 17, and then the rest of the provinces and territories had 6 or fewer cases. This is showing all of the indigenous outbreaks in this reporting period and the serotypes, which are predominantly type E. This makes sense given that type E is part of group 2, Clostridium botulinum, and they're found in aquatic sediments in northern coastline communities, and they also have a lower optimum growth temperature that permit growth in northern climates. This is a breakdown of all of the implicated food sources for foodborne botulism cases from 2006 to the present. Each case tells a different and interesting story. The first section is commercial retail foods, where there are some interesting standouts, including an international outbreak of carrot juice in 2006 that affected 2 people in Canada and 4 people in the United States. In 2009, there was an outbreak from ground beef that was left out at room temperature, affecting a husband and a wife in Ontario. In 2012, there was an outbreak from traditionally prepared uneviscerated salted fish called vesicle that affected 3 people. And in 2021, there was a case of a vegan Alfredo sauce sold in Quebec that affected 1 person. And temperature abuse was also the main factor in this outbreak. Home-prepared foods are not often implicated in outbreaks, which is actually good to see, considering the inherent risk of foodborne botulism from home canning or home jarring. But we did have some outbreaks involving a spaghetti sauce in 2006 and a watermelon jelly in 2011. And then, as I mentioned before, traditional indigenous foods comprise a large portion of outbreaks, and are caused by aged or fermented marine mammal meat. They're almost exclusively type E. And then finally, for many outbreaks, the food source is unknown. And these are categorized as unspecified foods. This is because the toxin was not found in the implicated food, sometimes despite the presence of viable Clostridium botulinum spores, which, as a reminder, are ubiquitous in nature. But that doesn't necessarily mean that the bacteria grew and produced toxin in the food that was consumed. So these cases could be due to colonization botulism, which is when the ingestion of the spores, when the person ingests spores and they colonize the gut and produce the toxin in the intestines. The low success rate for food origin tracing might be because most of these outbreaks were sporadic, that only included one individual affected, and most had no food samples submitted for testing at all. Outbreaks with multiple individuals linked by a common food make this sort of tracing much easier. In order to determine clinical outcomes of laboratory-confirmed cases of foodborne botulism, we retrieved patient records from the Canadian Institute for Health Information. These include the 2005 to 2021 Discharge Abstract Database and the 2005 to 2010 Hospital Morbidity Database, which is specific to Quebec. We queried all records that are available that listed botulism in the suspected diagnostic codes, and then we matched these records to our laboratory-confirmed cases. From this, we obtained 52 laboratory-confirmed cases that cross-referenced to hospital records. So in this reporting period, the average age was 57 years old, and 52% of patients were female. The average length of stay was 48 days in hospital, with a minimum of 1 day and a maximum of just under 500 days. 38 patients were transferred to special care, which can be considered the same as an intensive care unit, where they spent an average of 36 days and a maximum of just over 400 days. You can see from these graphs that the data is quite skewed by certain individuals who had significantly more severe clinical outcomes than others. 70% of patients required the use of a ventilator. Most of the patients were discharged to home with no support. 8% were discharged to home with some support from healthcare workers. 8% were transferred to continuing care. 17% were transferred to acute care, and 2% were transferred to palliative care. And then notably, 7 of the patients, or 13% of them, died. There have been some reports in the past of different serotypes having different levels of clinical outcome severity. So we decided to test this because we had the data available. And we did an ANOVA on the 3 serotypes that we had more than one case available. So that was A, B, and E. And we found a significant effect of serotype on both the length of stay in hospital and the time in special care. Then a Tukey's post hoc comparisons test indicated that cases of type A had significantly longer hospital stays and longer time in special care than types B and E. And interestingly, type A is the serotype that is used for the Botox injections for cosmetic and therapeutic purposes. So switching gears a bit, I'm going to now talk about infant botulism, which is a disease that affects children less than 1 year of age. There are about 4 cases per million live births in Canada, and they always occur sporadically. From 1979 to 2021, the average hospital stay was 20 days, and 35% of patients required mechanical ventilation, but there were no deaths reported. The cause of infant botulism is ingestion of Clostridium botulinum spores from food sources such as honey or environmental sources such as dust, and the production of the botulinum toxin in the gut of the infant after it colonizes the intestines. Children older than 1 year of age or adults have a more mature gut microbiota, and they can ingest Clostridium botulinum spores without colonization. Symptoms generally include what's described as floppy baby syndrome, which includes constipation, hypotonia, lethargy, weak suck, and weak cry, as well as shallow and rapid breathing. And again, treatments are very similar to adult. They're limited to mechanical ventilation and use of the botulism immune globulin, or baby big, antitoxin that is available from the California Department of Public Health. Today, I'm going to be showing you results from another publication of ours in the Canada Communicable Disease Report. This is a summary of infant botulism cases from 1979 to 2019, but again, I updated the figures to include more recent years. So this is the annual incidence of foodborne botulism in gray that I showed you before, and infant botulism in blue, dating back to 1960. The first reported case of infant botulism in Canada was in 1979. And when we compared the incidence of foodborne botulism with infant botulism, we found that since 2015, infant botulism has become more common in Canada than foodborne botulism. This is similar to the United States, where infant botulism is by far the most common type, especially in California. From the very first case of laboratory-confirmed infant botulism in 1979, there have been 79 cases with an annual case rate of about 1.8 per year and an incidence of 4.2 cases per million live births. This is the annual incidence of infant botulism separated by botulinum toxin serotype. The vast majority of these cases are type A in blue, followed by type B in orange. This makes sense considering that type A Clostridium botulinum are part of group 1, that, if you recall, have a higher optimum growth temperature, which would be better suited to growth inside the intestines than group 2. And of the type B Clostridium botulinum that were isolated, they were all identified as group 1. So it's almost exclusively type 1 Clostridium botulinum. We also had one case of a type AB and then a single case of a type F, which was actually identified by California Department of Public Health as being a Clostridium baradii and has been reported before. When we examined the age of onset for these patients, we noticed a unimodal distribution with a median of 16 weeks of age and a mean of just under 18 weeks. The majority of cases occurred before 26 weeks or 6 months of age. And this also corresponds well with previous studies. And it correlates with the typical age of weaning from breast milk or formula onto solid foods, which can cause changes in the gut microbiota and allow Clostridium botulinum spores to colonize the intestines. Of interest, the youngest case was from our single case of type F Clostridium baradii, and the older cases are all type A. This is also consistent with recent publications showing that on average, type A cases were older at onset than type B cases. So it's always nice when we analyze data and it agrees with the published literature. This is the geographic distribution of infant botulism cases in Canada. Of interest is the separation of type A cases in the western provinces and the mixture of type A and B cases in the eastern provinces. And this also, this matches the natural geographic distribution of Clostridium botulinum spores in the soils of the United States, where type A spores are more commonly found west of the Mississippi River, while east of the river, a mix of type A and B spores are found. To investigate the clinical outcomes for patients with infant botulism, we cross-referenced our records to available databases from CAI, similar to what was done with the foodborne botulism. And we matched hospital records for 23 cases. The average length of stay was 20 days, with a minimum of 8 and a maximum of 34 days in hospital. 13 of the 23 cases were transferred to special care, where patients spent an average of 12 days, with a minimum of 4 and a maximum of 22 days. Only 35% of the 23 patients needed mechanical ventilation as a treatment. The metrics on discharge disposition revealed that 39% of patients were discharged to home without support, 43% were discharged to home with the support of healthcare workers, 13% of patients were transferred to acute care, and 4% of patients, the outcome was unknown. And of the 23 cases, there were no reported deaths. I want to highlight some ongoing challenges for infant botulism, mainly that the source attribution is rarely determined. In contrast to foodborne botulism, in which multiple patients can be linked to the same food source, infant botulism outbreaks involve single cases that always occur sporadically. This is because Clostridium botulinum spores are ubiquitous in the environment, and spores merely need to be ingested at a time when an infant's gut microbiota is disrupted. So in the history of infant botulism in Canada, only 5% all cases were associated with a lab-confirmed source of spores. And these were all honey, which is already a well-established source of spores. And is commonly known not to feed infants honey nowadays. So we're still looking for the source of spores for 95% of these cases. The question is, where are infants being exposed to Clostridium botulinum spores? So this is a list of all of the lab-confirmed associations to date of infant botulism cases around the world to environmental sources, but excluding honey. Isolates of Clostridium botulinum from infant stool have been linked by serotype to isolates from environmental sources, including most commonly outdoor soil and indoor dust. It's possible that the isolates of Clostridium botulinum recovered indoors are simply from shedding of the infant's stool. However, one study in 1985 by Sarah Long and colleagues from Pennsylvania isolated Clostridium botulinum from soil from the father's work site, soil from the father's work shoes, soil from the household yard, vacuum cleaner dust, and indoor dust from the infant's room. So this demonstrated a clear pattern where spores from outdoor soils could be tracked indoors, where exposure to infants was most likely to occur. More recently, in 2014, Heidi DeBritz and colleagues from California used molecular epidemiology to match clinical isolates to the same genetic clades as isolates from the local surrounding environment, strongly suggesting that these geographic areas are reservoirs of Clostridium botulinum spores that lead to infection in infants. So this led us to ask the question, what is the prevalence of Clostridium botulinum spores in Canadian households? To answer this question, we collaborated with Pat Rasmussen's group in the Environmental Health Science and Research Bureau of Health Canada that were responsible for the Canadian house dust study. So if you haven't heard of this study, I recommend you look it up. It's pretty cool. The original survey was performed in the winter seasons of 2007 to 2010, and they were measuring heavy metals, such as lead. But the samples were still available, and they were being stored at room temperature, so we simply asked if we could use them, and it turns out that this lab was just down the hall from us in the same building, so it's pretty convenient to obtain these samples. We received 963 samples that were collected from 13 major cities across Canada with a population of over 100,000, and you can see them on the map below in the yellow stars. These dust were a coarse fraction of vacuum cleaner dust, between 80 and 300 micrometers. So it was a mixture of sand, dust, lint, and hair. Again, the results I'm showing you are from a recent publication in Epidemiology and Infection. And if you're interested in reading the details, you can look it up. And if you read the title of the paper, it's a spoiler alert. We didn't find any spores. But I think the results are interesting and worth discussing. And it's also encouraging that journals are still willing to publish negative results. We used a real-time PCR assay to detect the viable Clostridium botulinum spores in household dust. So very briefly, 150 milligrams of dust was weighed into sterile tubes and incubated in 3 milliliters of liquid media in an anaerobic workstation at 35 degrees Celsius for 24 hours, and then 30 degrees for an additional 24 hours. Then the culture was centrifuged and the pellet was resuspended in 100 microliters of TE and then boiled for 10 minutes and spun down again to collect the liquid. We used an internal amplification control for each well to ensure that there weren't any inhibitors from the dust affecting the reaction. And purified DNA from each type of Clostridium botulinum serotype was used as a positive control, and negative control was TE alone. Finally, to confirm that resident anaerobic bacteria in the dust samples were able to germinate and grow under these conditions, we randomly selected a subset of 3 dust samples from each of the 13 cities and streaked out the liquid cultures onto agar plates. Then pairs of unique colonies were identified by mass spec using a multi-biotyper from Brooker. The spectra generated were compared to a custom BDEL library, including 12,000 entries and a custom library of 324 strains of Clostridium botulinum. The results of this study are quite simple. 9 of the 963 vacuum cleaner dust samples produced enrichment cultures that were positive for PCR reactions for botulinum toxin genes A, B, E, or F. As I mentioned, maybe I didn't mention, but we randomly spiked a dust sample with a known quantity of Clostridium botulinum spores of each type, which were all successfully detected. We also had positive and negative control reactions for each set of PCR runs, and these were all successful. So the positive controls were all positive and the negative controls were all negative. Finally, the MALDI biotyper analysis revealed that Clostridium was the most common genus of the colonies identified, and Clostridium perfringens was the most common species identified overall. This may actually not be that surprising because the dust samples were being stored at room temperature for 15 years and cultured in anaerobic conditions. So the conditions were selecting for desiccation-resistant anaerobic bacteria. These results demonstrated that the liquid cultures did produce viable anaerobic bacteria of the same genus as Clostridium botulinum, and the PCR assay is a viable method of detection for Clostridium botulinum spores in dust. And so we suspect that the likelihood of false negatives in this experiment is low. The isolation of Clostridium botulinum spores from indoor dust and outdoor soils has been previously described. So why didn't we find any? One possible reason is that the dust was sampled from households of urban cities that could have a lower exposure to airborne dust than cultivated rural areas or suburban areas. The sampling was also performed in winter months. And in Canada, that usually means that everything's covered in snow, although not so much this year. So there was less likelihood of tracking in outdoor soils. The absence of recovered isolates could also be due to the simple fact that infant botulism is less common in Canada than the United States, and especially California. And the incidence may simply reflect the geographic distribution of spores in the surrounding environment. Individual cases could be an indication of a higher incidence of Clostridium botulinum spores in the surrounding environment. Therefore, thoroughly investigating all possible food and environmental samples from each instance of laboratory-confirmed infant botulism would help determine the origin of infection on a case-by-case basis. And speaking of which, as Paul mentioned, we recently started working with public health officials to try to follow up on lab-confirmed cases of infant botulism with extensive sampling of ingested foods and potential environmental sources. The two most recent were in October of last year and November of last year, where in the Simcoe-Muskoka District Health Unit, we had help from Shannon Kelly, and their team collected 11 samples, including 7 soil samples from around the household that were tested for viable Clostridium botulinum, and they were all found to be negative. Then in November of last year, Paul and his team followed up on a case of infant botulism with 13 food and environmental samples, again, all of which were found to be negative. So these results were discouraging, but I am thankful for the partnership with public health officials to help us to try to uncover potential sources of spores in food or the environment. Finally, I just want to let you all know that if you're interested in reaching out to myself or have any additional questions about botulism, there's a great resource available on the internet called the Botulism Guide for Healthcare Professionals. It's available on the Government of Canada website. If you Google search Botulism Guide for Healthcare Professionals, you'll find it. That's how I direct all of my calls with healthcare physicians and hospital administrators. I just tell them to Google it, and it always works. And if you want to reach me directly, my email address is richard.harris@hc-asc.gc.ca. And thank you for your attention. I'm happy to take any questions.
Lots of applause, Richard. Hope you can see that. I will say your presentation today has been fascinating, sobering, but extremely helpful. And I look forward to seeing some of the questions here. So I'm going to start at the top here. So this one, gee, I've put on my glasses. That's embarrassing. Wait a second. The problem with my glasses, if I put them on, I have to like get really close. And if I take them off, I have to sit back here. So I'll do my best. Bear with me. First question. First of all, Richard, thank you for the wonderful presentation. And Gelda may have missed it. For foodborne illness botulism, was there any specific interventions done to reduce the amount of indigenous related cases from the 75% to the 45%? Good question.
That is a great question. And the answer is no, there were no specific interventions. And the reason being is that we need to respect these indigenous communities and their traditional way of preparing foods. And so we didn't recommend any specific interventions like cooking the food, which would have been a simple one to destroy the toxin. But I think that just interacting with the communities, talking with them, following them, and working with the local public health up there to suggest that they store the marine mammal meat at a lower temperature, so in refrigerators or freezers instead of burying them outdoors, probably contributed to a reduction in the incidence. But it may also have just been through bringing awareness to the issue and working directly with these communities.
Very good. Thank you. Okay, and so from JSL, one case investigation had a mother giving infant home remedy of chamomile tea and honey. I think there was some study that showed chamomile could be a source for spores. Just wondering if that has ever come up.
Yes, I am aware of that that case study, and it's true that chamomile tea, they did find Clostridium botulinum spores in that one. So it's true, you're right. Yes, it's just not as common. So it's a one-off case, and so it's an example of following up on each individual case and trying to determine, what did they put in the baby's mouth, right? And it could be anything, really. Honey is the most obvious suspect, but it could be any number of things. So it's a good example of testing just about anything to see if the spores are there. And it was actually quite surprising, I think, that chamomile, that they found spores in the chamomile tea. So it's a great example of that. Thank you.
Thank you. Next question from Dr. Keith Warner. When looking at the prevalence of botulism, is it better to do for-cause testing, for example, or that is, botulism is likely to occur in anaerobic sludge that is deposited on land?
We have a mandate to test clinical samples and food samples when cases of botulism are suspected. So we're not involved in sampling from the environment and ecology, but it is an area that I am interested in. And I think that it would be great to do a wide-scale ecological study looking at the natural environment of Clostridium botulinum spores in Canada. It's something that's never been done before. In the United States, there were soil samplings from 100 years ago. This work was done. And so the data is quite old, but yeah, it was interesting to see the geographic distribution of the different serotypes of Clostridium botulinum in soil samples in the United States. It's never been done in Canada, and I would love to be involved in that.
Thank you. From Robert Reed, do you test any samples not associated with human cases?
It's probably animals you're talking about, so like birds or cattle, that sort of thing. We do not do any testing for animals. We have probably the capacity to do it. We don't have the antitoxins for the neutralizing antibodies for types C and D. And I'm not aware of any other lab in Canada that does testing for animals. So you might have to connect with some veterinary labs in the United States And I think I might be able to help you out if you want to send me an email.
Thank you. From Nagme, Public Health Ontario. Was there any information provided on proper canning of seafood that could have contributed to the reduction?
Seafood. I guess they're asking about the indigenous communities. So it was the traditionally prepared marine mammal meat is not canned. So it's typically aged in plastic bags or some other containers and buried in the soil. Although now it's recommended that it's done in refrigerators or freezers. So there weren't any, it wasn't a matter of improper canning of seafood that was contributing to the incidents. It was a traditional method of preparing the marine mammal meat that was having an effect.
Thank
I
you,
hope that
Richard.
answers your question.
Thank you. Next question from Matt Hurst. Excellent presentation. Thank you. Is any immunity conferred from previous infections?
Actually, yes. That's a great question, and the answer is yes. So recently we've been looking at iatrogenic botulism cases, although there haven't been any confirmed ones, just suspect. And iatrogenic botulism is botulism caused by Botox injections, so therapeutic or cosmetic purposes. So I've been doing some reading on that, and it's true that the more Botox injections you get, the less effective they become. And that's because our bodies naturally produce antibodies to neutralize the toxin as a foreign antigen. And so yes, previous infection or previous ingestion of the toxin does confer an immunity to follow up cases. So yeah, it's a good point.
Thank you.
Lots of questions.
Yeah, so the list keeps growing.
That's a good sign.
Yeah, very good sign. I've read that you should never defrost raw fish in their original shrink-wrapped packaging, as this could cause botulism. Can you help explain why? And that was from Kim Landers from Amafra.
Defrost raw fish in their original shrink-wrapped packaging. So the alternative would be to transfer it to a new container. So,
or maybe defrosting at room temperature in the shrink wrap versus the fridge. Yeah, not
the
quite
shrink
sure.
wrap vacuum package, the shrink wrap plastic vacuum packaging is a perfect environment to create an anaerobic environment for the growth of Clostridium botulinum. And there have been cases before of this happening. So yes, it is a risk to leave vacuum packaged fish out for too long because spores could be present on the fish, and given the right environment, an anaerobic environment, and a high enough temperature, they could start to grow and produce the toxin. I don't know if your question is asking whether you transfer the fish to a new package would help to reduce that. I wouldn't think so, because the spores would be on the fish and you transfer them to a new container, but it wouldn't be vacuum packaged anymore. So the risk of producing an anaerobic environment would be lower.
Thank you, Richard. From Brian Krause. And yes, actually, I recall this being an issue as well. Are baked potatoes wrapped in foil still an issue? I recall an outbreak in Ontario in 2002 involving temperature abuse and a baked potato wrapped in foil.
Baked potatoes, or potatoes in general, yes, are notorious for foodborne botulism. And as I mentioned before from the previous question, anytime you provide an anaerobic environment with the right conditions, then there's a risk of that. So yes, the answer is yes.
Thank you, Richard. Talil Marinko from WDG. There was a case you identified with both A and B toxins. Is the assumption that there were two different types of spores ingested, or that there is some chance of a single spore producing both toxins?
That's another great question. And the answer is that it was a single isolate that was producing both A and B toxins. And that's been found before. Some Clostridium botulinum isolates, they have both A and B genes to produce the toxins, but the B is silent. So they're an A silent B. And some of them have both toxin genes and they can express both. So we need a combination of A and B neutralizing antibodies to identify it. So yes, it is one isolate expressing both toxins.
Thank you. From Bruce Ferry. Wow, great presentation with appropriate data and analysis of the data. Interesting to learn that type E is a major issue in Canada. I heard a lot about the US state of California. Is California a major location in the US for botulism? Possibly because of the agricultural dust created with all the farming, perhaps, but what do you think, Richard? Is California a major location?
The answer is yes. California is the center of the world for infant botulism. And so I collaborate a lot with colleagues from the California Department of Public Health. It's where they developed the baby big antitoxin specifically for use in infants. So it might be a case of they're just more aware of the disease, and so that's why they have a higher incidence. But it is a region of the world that has the highest incidence of infant botulism. So whether it's created by agricultural dust or other factors, that is an active area of research. So you can expect to see publications coming out soon looking at this sort of thing, trying to determine the source of spores just like we are. And there's a lot of motivation to do that simply because infant botulism is so common in California that they're motivated to answer the same questions that we are, find the source of these spores.
Couple more to go, Richard. So from Christina Lee, PHO, thank you for the informative presentation, Richard. Is there an explanation for the increase of infant botulism cases?
Yeah, I think it is partly due to awareness. So the first case of infant botulism was in California in 1976. And then in 1979, the first case in Canada. And then since then, it's become more, it's been more made more aware. And so I think it has partly to do with that. Before 1976, there were probably cases that went under the radar. And there's an interesting connection between infant botulism and SIDS, so sudden infant death syndrome. And that is that the age of onset, that unimodal distribution that I showed before, where it's most common at the age of weaning off of milk or formula into solid foods, that unimodal distribution of age is almost exactly mirrored by SIDS cases in infants. And so it's been speculated in the past that there's a certain proportion of SIDS cases that could be attributed to infant botulism. It just wasn't, people weren't aware of that possibility before the late 1970s. So, yeah, it's a great question.
Final question, Richard. Is there a time? Oh, no, I lied. Two more to go. We have time for these two. Is there a time frame with illness when immune globulin is not effective?
I'm assuming they're meaning the antitoxin.
The antitoxin, yeah.
Yeah, there are some really good publications on the effectiveness of the antitoxin, both Baby Big and the heptavalent botulinum antitoxin. And the window of opportunity for the maximum effect, I've heard, is something like 6 to 8 hours. And then after that, the effectiveness wanes. But it's always better to just treat a patient with antitoxin anyway if the symptoms are so severe. And so that's typically what is done. So if a patient presents perfect symptoms of botulism, then the doctor will just recommend the administration of the antitoxin, even if it's a day, 2 days later, it still has some effect, and it still will help with recovery.
Thank you. Can you speak more on removing or inactivating preformed toxin in food using heat?
So destruction of group 1 spores is a challenge. It takes extremely high temperature, so ultra high temperature treatment, or a combination of pressure and temperature. And so in general, Clostridium botulinum group 1 spores are considered the most difficult microorganism to destroy in foods. So if you can destroy them, then you can be pretty sure you're destroying everything else, especially gram negatives. So that 12D process, that botulinum cook that I was talking about before, that is essentially, if you can achieve that, then your product is basically safe to store at room temperature, which a lot of we're looking at non-dairy milks now, which are treated in a similar way, and they're all stored at room temperature on the shelf in the grocery store because they've undergone a bot cook. So they're safe. They're essentially sterile. The toxin is heat labile. So while the spore is difficult to destroy, the toxin is very easy to destroy with temperature. So if you cook your food, you only really need to reach a temperature of like 70 degrees to destroy the toxin. So in general, it's better to cook all your food. Preventing spores from contaminating your food or destroying them is challenging, but cooking your food after is relatively easy. And that's just one of the areas that we don't really get involved in is public awareness and public education and that sort of thing. So it's an area that I would like to explore and work with others in best practices for storing foods, preparing foods, best practices for that sort of thing.
Okay, thank you. I think we can squeeze in one last question, I think, and this is from Alan from Hamilton. Why is Baby Big so expensive?
You'll have to ask California.
Okay.
I would assume it's because they're the only lab that produce it. It is expensive to produce because it's made in humans, and so it's not equine derived. And but it is effective. It is the most effective for infants. So there has been some talk in the past about treating infants with the botulinum antitoxin, the heptavalent one that is usually used for adults. And we've found that it's not as effective in infants. So it is recommended to treat infants with Baby Big.
Okay, thank you. Okay, so that concludes today's seminar. I do want to see a point here from Lil. Thank you very much for the great presentation. It was fantastic working with you. I think we can also thank Lil, because Lil, after working with you, Lil said, Paul, we got to get Richard in for an in-service. And so, of course, why just do an in-service? Let's do an out-service and do it through GFFS. So thank you, Lil. Lots of applause here. That is awesome. While everybody is in a mood to applause, we're on a mission, I think. Let's help Richard on his mission to find that isolate. So I encourage everybody to, when you do have a case like this, make the effort to go out and try to find the isolate. Take the samples as well. It's well worth it. I'd also have to say, let's please rely on Richard for guidance and support in any cases you have. He was a wealth of information for us, a great person to have in your Rolodex, if anybody still knows what that means. Finally, I'll say that the meeting has been recorded. If you would like a copy of the presentation, and I think you should get one and make it part of your orientation for all new personnel who are working at your agency. It's a great primer for anybody on your team. Please get a copy of that. So at this point, Richard, once again, can't thank you enough for your time today. And I thank everybody for coming on as well, asking great questions and doing our best to improve food safety and infant safety in Canada. Thank you.
Thanks, Paul. It was great working with you too.